live. So, good morning, everybody, and thanks so much for joining us for our first Fireside Chat. So I'm joined with GenXys, is Andrea Paterson, who's the algorithm team manager on the team at GenXys, and she's a practicing clinical pharmacist at an interdisciplinary primary care clinic. She's completed her Bachelor of Pharmacy and postgrad community pharmacy residency at UBC in Canada. And she has specialized training in palliative care and chronic pain management. And she also lectures at the UBC. So she sounds pretty busy. So thanks for making time for this webinar. And before we get started, I just want to explain how this will work a bit. So Andrea and I are going to chat for about 30 minutes. Then we'll answer some questions at the end. So we have some moderators, Shay and Christina and Kathleen on as well. And so they'll be tracking your questions in the comments section, and we'll do our best to answer some questions. So today we're talking about improving treatment outcomes with pharmacogenomics. So if you have some kind of support question for GenXys or DrugBank, it's probably better to send that to the support channel, not the webinar, not this webinar. So, the other thing to note is that this webinar is going to be recorded, and so it will be available after to rewatch following the session. And with that, we'll just dive right in. So how are you doing today, Andrea?
Great, thanks. How about you, Mike?
I'm doing very well. Yeah, so I gave you a little bit of an intro, but it'd be great to hear you tell us a bit about yourself.
Sure, great, thanks. Well, yeah, great to be here. So my name's Andrea Paterson, as you said. I'm a clinical pharmacist, and I've been with GenXys Healthcare Systems for about 5 years now. And my role there is algorithm team manager. So I think we'll talk about that a little bit later about what we do. But basically, our team creates the content for our clinical decision support software, TreatGX. And then I also work part-time, as you said, in a primary care clinic as a clinical pharmacist. And that's really important to my work at GenXys because our vision is to power every prescription with our software. And so in order to best do that, we need to be up to date with the most current practices so that we can make it as helpful as possible to patients and healthcare providers to use the software.
That's excellent. And can you tell us a bit more about GenXys?
Sure, yeah. So we created our software, TreatGX, with the goal of improving medication safety, increasing medication efficacy, and also reducing healthcare costs. So those are some big goals, but we really felt that we could do this through more personalized prescribing and giving more personalized medication treatment options to prescribers so that they can better help their patients. And so basically our software works by using evidence-based algorithms to offer a list of medication treatment options for a condition. And those options are basically personalized for each patient whose information is entered into the software. So that uses so many different variables. So pharmacogenetics, we also do a pharmacogenetic test, is one of those variables. But the other really important variables that need to be included are other things like kidney function, drug-drug interactions, drug condition interactions, other lab values. So it's a ton of information that we're trying to kind of funnel into the software to put at the fingertips of clinicians who can really use it to improve patient outcomes. And then our pharmacogenetic test is really exciting as well because that allows us to pull in even more information for personalization. So in Canada, we've worked with Life Labs to offer our test across Canada, and then the software is now used in major pharmacy chains and clinician offices, and also by some big health insurance companies like Sun Life in Canada. And then we just recently started expanding more into the US, and our software is used by some early adopters there, which is really exciting.
Excellent. Yeah. So you're mainly operating in Canada right now. I know we've got some people on the line from different countries. Mm-hmm. And when did you get started with GenXys? It was, what year was it?
So 2015 was when I started with GenXys. Yeah.
Okay. Nice. Yeah. Nice. That's great. And so it'd be great to hear a bit more about your role as an algorithm team manager. It's a pretty cool title.
Yeah, it's a unique title for sure, but I think it kind of encompasses what we do. So we have 3 main teams that work to kind of power and create our TreatGX software. So we have our pharmacogenetic lab team, and so they're in charge of all of the pharmacogenetic panel development. So figuring out what are the best pharmacogenetic tests to include on there, which ones are going to be the most clinically relevant and actionable. And then they also look at the gene-drug associations in terms of us integrating those into the software. Then we have our software engineering and health technology design team, and so they basically design, build, and maintain our software. And then my team is the algorithm development team. So we are generating the content for the software. So what we're doing is we're researching and creating these best evidence treatment algorithms using a whole bunch of different sources. So looking for the best levels of evidence in terms of the newest research, but also looking at treatment guidelines, kind of trying to distill everything down into this algorithm that's based on the best clinical evidence available for treatment of a specific condition. And we have about 38 conditions in our software right now. And then once we have that list of medications that we know are going to be, generally safe and effective to treat that condition, then we have to take all the different patient variables that can occur and bring that into medication selection even further. So maybe excluding some medications that might not be safe, and then also fully dose adjusting all of the medications. So my team has to sift through a lot of information on a daily basis to try and find the most relevant and important information and really make sure that the medication treatment options that we're presenting in the software are going to be as personalized as possible so that patients can have ideally the best outcomes.
Yeah, that's really interesting. It's great. It's always great to learn about some of the different things that our customers are doing. We have lots of different types of use cases, and this is a particularly cool one, I would say. And so how does DrugBank fit into some of the things that you're doing?
Yeah, so I think we use several different data sets to help integrate the vast amount of data that we use in the software. So the big one for us is the drug-drug interactions data set because clinicians don't want to use our software to get medication options, but then have to go to another software to look for drug-drug interactions. And as I mentioned earlier, drug-drug interactions do play a big part in selecting the safest and most effective medications for a patient and trying to avoid adverse drug reactions. So definitely the drug-drug interaction database allows us to easily integrate that information so we can look at the treatment options we're offering to a patient and then look at their current medication list to make sure that any of the medications we're suggesting are not going to be interacting in a really impactful way with their current medications. And then we also use a variety of other data sets like the prescribable name search to allow patients to enter their medications, and then we can then store those medications and the properties that we want to be able to use in a very structured and searchable way. So then we can really use all the information that they're giving us to help further tailor those medication treatment options.
Great. Yeah, thanks for that detailed answer. That's awesome. So you're looking at quite a few different data sources, and what are some of the things that you look for when you're looking at the quality of that data source, and how do you kind of make those decisions on what to include?
Yeah, so I mean, the quality and the personalized output of our software is only as good, obviously, as the data that goes into it. So a big part of my team's role is doing a lot of critical appraisal when we're looking at data. For the drug-drug interaction database, for example, we were looking for something that was going to be really structured, evidence-based, up-to-date is really important because a lot of information that you can sometimes get in drug monographs is from 20 years ago and it's never been updated. So that's really important to us. And then we also are obviously constantly growing as a company, and our software is kind of expanding its reaches. So we're always also looking for information and data sources that kind of are scalable and also have multi-country data, because initially when we were just in Canada, we just had to look at, we were looking at the best evidence for medications, but we're looking mostly at Canadian monographs and, considering Canadian guidelines. But now that we're more international, we have to look at a lot more comprehensive information and take into account not only practices here, but medications that are used in other countries as well.
Mm-hmm. That's great. Yeah, and I guess it'd be interesting to talk a bit more about some of the interesting trends that you see in the field of pharmacogenomics. I mean, there's a lot of people talking about testing with COVID-19, and there's a lot of innovations in the testing area. So, and I think people are really starting to think differently about their health information because of what's going on. So, yeah, it'd be great to kind of dive into some of the trends that you've seen as an expert in pharmacogenomics.
Sure. Yeah. So yeah, definitely COVID-19. That crisis has definitely, I think, changed the whole field of medicine and really accelerated that move to virtual health and more digital tools. And so I think people are looking for more digital prescribing tools to help them deal with all of this different information. And there's so much information at our fingertips that we can use. But yeah, integrating that is really hard. So in pharmacogenetics, traditionally, a lot of tests that patients are getting done will just give you a paper report. And so patients will bring that into their clinician or to their pharmacist, and here you go, here's a 50-page report. And that's not really usable in a clinical setting because no one has that much time to go through it and to figure out, what applies to what drugs. And even if it says, what drugs are affected, what happens if you also have kidney impairment? How do you integrate those two variables together? And also, a lot of times medications are used for multiple conditions. So if you have, a reduced dose for medication in one condition, that might be a very different dose than used for another. So I think what we were really trying to fill the hole of was, that lack of interpretation of the pharmacogenetic information into a clinically usable way. So having the TreatGX, automatically pipe in pharmacogenetic results and then use that with all of the other important information. That's really big. And I think one of the big barriers to implementation of pharmacogenetics in clinical practice is really knowledge of clinicians. Many don't feel confident enough to use that information, but also time. You just don't have enough time to sort through for each patient who brings in one of those tests, what kind of implications that might have for their future medication therapy. So you really need those automated ways of storing that information and then using that information when you're prescribing. Because you're right, I think many people are getting pharmacogenetic testing done. Cost is coming down for it. There's much more recognition that for many medications, it can significantly affect response and also risk of severe adverse side effects. So I think there's starting to be more recognition of that. More care centers are starting to use pharmacogenetics. The FDA has recently published their list of pharmacogenetic markers that have an association with certain drugs. So I think the ball is definitely rolling, but it's just a matter of finding the best way to implement that in clinical practice.
Yeah, that makes sense. And there's a bit of a learning curve for the adoption of it, I guess, as well. And then you mentioned that, so you work with a lot of pharmacies, but you also work with insurance companies. So what are the trends in that field?
Well, I think so obviously insurance companies, they want to help the patients that are under their plan have better health outcomes because that benefits everyone, the patients and the insurance providers. And also, I think reducing costs is a big thing. So a lot of the prescribing currently is kind of trial and error or one size fits all because a lot of studies are looking at, a wide range of people, and it's really hard to narrow down, for this person, this specific drug is going to be effective. And in mental health, I think that's one of the big areas where, we have a lot of evidence that, for example, the SSRIs seem to be pretty effective in most people. But we kind of look at them almost as a class when we really need to be looking at them more as individual medications. So what are the different factors that affect each of those medications in the class? How do pharmacogenetics play a role? How does kidney function play a role? What are the side effects of each medication that patients, are unlikely to be able to tolerate? Or maybe there's a side effect that might benefit them. So if they're having trouble sleeping, maybe a more sedating medication would work. So I think insurance companies really recognize that they want to try and get people on the right medication right from the start. And a software like TreatGX that integrates all of the variables you need when looking at the person as a whole, I think really helps move in that direction.
Mm-hmm. Nice. Yeah, and I guess AI probably plays an important role in some of this as well. So could you talk a bit about kind of how does AI play into your role or the things that you're doing at GenXys?
Well, actually, so it doesn't right now for me, but there's
Okay.
lots of really exciting opportunities, I think, in the future for that. So there's so many things that we'd like to use AI for. So looking at, for example, searching the literature and trying to select, relevant information, because for us, that's a big part of our job, is keeping up to date with all the new research studies and clinical guidelines that are coming out. So if you had AI integrated in a way that could keep track of that and flag important papers, that would be really useful. But also, I think one of the big things in the personalized prescribing realm that we're trying to pull in is patient outcomes. So using patient-entered data, so tracking their own health outcomes, so whether that's side effects or outcomes from medications, and feeding that back into our software so we can start to get this picture of, certain types of people are more likely to respond a certain way to medications, so that we can build kind of an adapter algorithms to be really effective for patients, improving the outcomes that they get from their medications.
Nice. Yeah. So it's something in the future that will always be improving and improving as you get more and more adoption.
Exactly.
Yeah. So, I mean, you touched on this a bit, but what are some of the really interesting or exciting things that you've seen happening in the field of pharmacogenomics?
Well, yeah, I think just the wider expansion and acceptance of it, kind of moving. There's been, some really great articles looking at, are pharmacogenetics going to become the standard of care? Will it be, neglectful if you're not doing that for patients, especially for medications that are really affected by them? And I think we're kind of, on that cusp of, a lot of clinicians will have this pharmacogenetic data about patients but not know how to use it. And so I think we really need to put tools in their hands so they can use it, because if you have that information and you're not using it, you're really, it's like you're ignoring a lab value for the patient. It's all valuable information that can go into prescribing. And then I think the other thing too is, we've known for a while what are some useful pharmacogenetic tests in terms of, we really know that this specific pharmacogenetic variant is associated with this kind of outcome. So we know, you might need a lower dose of a medication, or you should avoid that medication, or you might actually respond better to that medication. But one of the really tricky questions has been, when do you do pharmacogenetic testing and who do you do it on? So I think there's also been a move to try and identify patients who might be good candidates for pharmacogenetic testing. And often right now, those patients seem to be patients struggling with mental health issues. So they've tried, a antidepressant or two and had not had much benefit or had intolerance to them. So trying to do pharmacogenetic testing before they move on to a third and fourth to see if you can kind of narrow down a little bit more what medications might be most effective. And then also in cardiovascular health, we know that there's a lot of pharmacogenetic variants that affect medications used to prevent long-term outcomes. And so therefore, if you can do testing earlier and get them on the right medications, you can also prevent adverse drug events. And I think there's much more recognition now that adverse drug reactions are a leading cause of death, and considered to be within the top 5 leading causes of death in many countries. So trying to reduce inappropriate prescribing, getting people on the right medication earlier on will really bring down healthcare costs, but also help patient outcomes, which is the goal.
Yeah. And the tools that you're making are so important for that problem, because the most complex patients are the most likely to have these issues and the hardest to understand. It's the more difficult to use the data about that patient because they're more complex. So your tools can really help a lot with just understanding all of that data and actually making use of it. So that's really exciting. Yeah, and I guess you touch on that this next question a bit. So, what are the most common issues and opportunities you see with treatment outcomes? So I guess this mental health, that's one that's a really exciting opportunity. But is there, what's the most common case that you're seeing with your service?
Well, definitely, I think, yeah. So a lot of people who've had a lot of bad reactions to medications. In clinic, I see a lot of patients who say they're sensitive to a lot of medications, and before you kind of just say, oh, well, that maybe that's just your mindset. But we actually do anecdotally see that some patients, who have one variant for medication actually have a lot of different variants. So it makes sense that they might be more sensitive, especially if they have a variation in one of the genes that affects a lot of different medications, like P450 enzyme variants. Those ones can have a lot of different effects on medications. And I think it's interesting because we have so much information at our fingertips. There's been this huge move to evidence-based healthcare over the last 20 or 30 years. But the evidence is often, as I said, kind of class-based and not really specific to an individual patient. We're looking at a population in a clinical study. So you really need to start to use that AI and other kind of N of 1 studies to look at, for this particular patient who has, these pharmacogenetics, this kidney function, these conditions, these other medications, this is, a likely outcome for them if they take this medication. And we're not quite there yet, but I think with, more data and more ways to integrate that into prescribing, it will start to get better, hopefully.
Yeah. And you mentioned the P450 enzyme. So what's an example of, I guess, why does that affect lots of drugs? Just to kind of, for the people watching, just to give a bit of detail there. So I think we had a few questions coming in about people are trying to understand exactly what it is. So that could be a good example.
Sure, yeah. So when you're looking at pharmacogenetic variations, so the changes in our genes that they might have an effect or they might not. In pharmacogenetics, so the study of genes that affect your response to medications, there's kind of two different types of markers we look at. So one is a pharmacokinetic variant. So that changes kind of the drug metabolism and how your body deals with the drug. So one really common example is CYP2D6. So CYP2D6 is an enzyme that metabolizes drugs either from active form to inactive form, or also potentially the other way around. So the most common, I think, example that many people know is codeine. So codeine itself does not have very much analgesic properties when you first take it, but in the body, CYP2D6 converts it into morphine, and morphine is much more analgesic in its action, so it has much more pain-relieving properties. So if you're a poor metabolizer for CYP2D6, you convert codeine into morphine much more slowly, and you're unlikely to get as much pain relief. If you're an ultra-rapid metabolizer for that enzyme, you're converting codeine to morphine really quickly, and so people often get this kind of high, or they feel much more effective and much more quickly. And so you can have, two people who look exactly the same, might have the same kidney function, kind of exactly the same, but if they have different variations in their CYP2D6 enzyme and it can really affect how they respond to their medications and can have potential severe adverse effects. So codeine is recommended not to be used in breastfeeding mothers anymore because some mothers early on would take it not knowing they were ultra-rapid metabolizers, and then they would have a very large amount or a high concentration of morphine in their body, which could then be passed on to the breastfeeding infant. And I think there have been a couple case reports of infant deaths due to that. So that was one of the big kind of, this is a really important enzyme. This can really affect outcomes. So now looking at these enzymes ahead of time, like CYP2D6 probably is responsible for metabolism of close to 30% of the medications that we use in primary care. So it's a really important one to know and can affect the doses of medications or which medications you might choose to use.
Yeah, that's a great example. Thanks. And I think some people were also asking how would a drug interaction affect the pharmacogenomics? So maybe since we're talking about the details,
Sure.
answer that question now as well.
Yeah, let's throw that in there. So there's a couple different ways. So one thing that we've been looking more into is something called phenoconversion. So that would be, someone who might be a normal metabolizer for CYP2D6, but some medications also alter that same enzyme pathway. So you might have a medication that is inhibiting the activity of CYP2D6. So even though you're a normal metabolizer, if you're taking a medication that causes a reduction in the CYP2D6 activity, your body is acting like you're a poor metabolizer for that enzyme. So that's one way. The other way is just two different effects potentially on the same medication. So if a medication is metabolized through two pathways, so say it goes through CYP2D6 as one enzyme, and CYP3A4 is another really common enzyme that's used in our bodies to metabolize medications. So if you are a poor metabolizer for CYP2D6, the recommendation might be to reduce the dose by half. But then say you're also taking a medication that inhibits that CYP3A4 enzyme, then you don't have really any metabolism potentially left of that medication. And so you might want to avoid that medication completely rather than taking a lower dose. So that's kind of where the drug interactions come in. Mm-hmm. If you have one drug that you're currently taking that affects generally the metabolism of another drug.
Perfect. That's great. That really makes it clear. Yeah, I guess going back to kind of your work at GenXys, so what's kind of the most game-changing experience or the most exciting thing that you've seen happen while you're working at GenXys?
Yeah, so I think when I first started, it was more, for me, it seemed more of an academic pursuit because we weren't live yet. We weren't doing pharmacogenetic tests for patients, but as soon as, we went live and we started doing pharmacogenetic testing, to hear patient stories and clinician stories about how it actually helped them. So we have lots of examples of patients saying, I tried multiple medications, I wasn't having any luck. And then, I did your test, I used the software and I started this medication and it really helped. Or, other cases from healthcare providers who said, I had this patient, who was taking a medication called clopidogrel, which is metabolized through that same enzyme system, but through CYP2C19. To its active form, so it goes the other way. And this patient was on it after a heart attack, got a clot, and had to have that clot then removed and a new stent put in. And so by testing, he was figured out to be a poor metabolizer, so he actually wasn't getting the protective effect from that medication that he was expecting to get. So then they were able after that to put him on a different medication. So just real stories like that, where you're actually seeing the impact in everyday life, I think that is super rewarding. And it really tipped the scales for me in terms of, wow, like we can really make a difference and we can, continue to improve the software so that we really are kind of, on the cutting edge of what you can do to personalize prescribing.
Yeah, that's great. That's really exciting. Yeah, you're having a big impact on people and improving their treatment outcomes, even saving lives sometimes. So that's really exciting. And for me, it's, something that's exciting for me to see is companies like GenXys that are really helping to take all of this knowledge that we've built up, scientific knowledge, knowledge about medications, and really use it to have that direct impact on people. And it's so it's really neat to see what you folks are doing. I think that we're going to go to some questions now. Okay. So some of our moderators have been collecting questions. So Shay, you got some for us?
Yeah, we'll start with Lucy's question. Lucy asks, I was wondering what kind of lab test results of patients are entered into the GenXys software.
Sure. So kind of the two most common that we use for most conditions, one would be your kidney function, so an eGFR. We look at our serum creatinine. So many medications are affected by kidney function in terms of metabolism. So that's the most common. Another factor that we look at that's not purely from a lab would be liver function or liver impairment. and we use the Child-Pugh scale for that. That's the most common way of coding liver dysfunction in drug monographs. So that's what we use. But we also, depending on the algorithm, will use things like potassium level for hypertension. We'll look at INR if we're looking at atrial fibrillation with warfarin dosing. So those are probably some of the most common ones.
Awesome.
Great.
So the next question is from Chris. When it comes to obtaining a patient's profile, is it typically through a whole exome sequencing or some other method?
So our test right now is a targeted panel. So we're looking at, I believe, about 60 different pharmacogenetic variants. So another thing that's really important to us with the test is that, when people want a pharmacogenetic test done, they don't necessarily want all of the disease markers that come with whole exome testing. So some people just really want, something that's going to be actionable. So when we're selecting the markers that go on our panel, we want to make sure that they don't really have those disease implications. So genes like ApoE, which can predict response to cholesterol medication, also can predict your risk of developing Alzheimer's. And so for a lot of people, they don't want that. They just purely want the pharmacogenetic information that can be used for prescribing decisions, but don't want to know about those disease risks. So right now, we do use a targeted panel. So SNP, looking mostly at SNPs and some HLAs, as well as some copy number variants.
That's really cool.
Is there also kind of a cost factor when you're looking at these different potential ways to test, exome sequencing versus panel?
Yeah, exactly. Whole genome and whole exome sequencing are more expensive right now, for sure. The targeted panels are generally more affordable for patients and also more accurate in some cases. So some genes are really hard to really pull out the true genotype from looking at just the whole exome sequencing, especially if there's copy number variations. So really, you almost, if you're taking whole exome or whole genome data and translating that into pharmacogenetic information from some of the markers, sometimes you do actually need to do some targeted testing to really clarify some of those variants. So for pharmacogenetics, it seems mostly it is the targeted panels that are used right now.
Awesome. All right, the next question comes from Ashok. GenXys looks exciting and promising. My question is about the role of microbiome in personalized medication. Do you think we can use microbiome information to predict the fate of drugs? Is there any specific input formats for pharmacogenomics data?
So microbiome, is something we don't do a lot with right now. I think that's a really interesting field, and I think there's more and more data showing that definitely, all of the microbes all over our body can play a big role in health as well as potentially medication response. But right now, that's not something we integrate, just because I think in terms of making medication decisions based on that, the evidence is not really there. What we look for in markers that we include on our panel are studies looking at people with a specific variant and then a specific response they get to the medication that might be different than normal. So it can be really difficult with some markers to find that. It's much more straightforward with markers that are involved in metabolism, because you can kind of see sometimes a linear correlation between, if you're a poor metabolizer, you have, this much drug, circulating in your body, versus if you're an ultra rapid metabolizer, and then the effect of that is this. So there's some markers that are less able to kind of elucidate, more effect versus less effect. And I think the microbiome is one of those things we just don't really know how to use that clinically right now.
All right. So our next question comes from DP. How did you select which diseases to include as part of the software?
Yeah, that's a great question. So a number of different factors. Initially, we were mostly looking at wanting to cover some of the most common primary care conditions. But we've also expanded now to look at some secondary care. And it's kind of a mix. So it's common conditions. So we wanted to ideally cover, the most common conditions that could be seen when you go see your family practitioner, and then are starting to expand a little bit more to secondary care. Some of our algorithms actually don't have any pharmacogenetics in it if there's no pharmacogenetics that are relevant to the medications used to treat that condition right now. So some of them are purely just clinical decision support softwares that incorporate, drug-drug interactions, conditions, lab values, and best evidence. And then others we've chosen because there are really valuable pharmacogenetics that can be used to help guide treatment.
Great. So I'm going to switch it up with this question, and this can actually go to both Mike and Andrea. Amelia asks, I hope you are all staying safe during this quarantine. As a pharmacy student, I was wondering how you decided on your career path and what steps you took to get there, as it's not a traditional community or hospital pharmacy kind of job.
Yes. Should I take us? I'll start that.
Yeah, you start. Yeah.
Sure. So I think like many people who go through school, I didn't really know what I wanted to do when I finished. I had learned about pharmacogenetics in school, but honestly, it wasn't really on my radar because it kind of seemed like more of a research thing when I had learned about it. But I was really interested in primary care and that patient contact. So I actually did a postgraduate residency, and during that year of residency, I was able to rotate through a whole bunch of different sites and learn more about what kind of practice settings I was interested in. And then one of my advisors in the residency actually connected me with GenXys. So it was a little bit of luck, but also hard work, I think, that led me here. But I think one of the best things that you can do as a student is just try as many opportunities and as many workplaces, I think, as possible. I worked in a couple different pharmacies going through school, and then residency gave me that opportunity to kind of rotate around to different sites as well. So I think just trying out as many things as possible and making as many connections as possible will help you kind of find your niche later on.
Mm-hmm. And when you're in university, there's lots of opportunities to try out different things, meet different people. So you have to take advantage of that. We've hired quite a few pharmacists as well. So look at our careers page if you're a pharmacist looking for jobs. Little shameless self-promotion there, but there's so many different options when you have a pretty much any degree. You kind of imagine that one specific job when you go into that program, maybe, but if you keep an open mind and you try to experience these different opportunities, there's a lot of exciting things happening in the world and lots of innovation. So working at a startup can be a pretty challenging and rewarding experience, and a great way to use your skills and learn a lot of new things at the same time. So definitely, if you're a student on the line, probably take a look at some of these other types of opportunities, not just the traditional role. And look how big of an impact you can have when you work at a company like GenXys, where you're actually impacting tons and tons of people with the product. So that's an awesome question.
Okay, so our next one comes from Dundi. Is there any methods for the implementation of pharmacogenomics in public health?
I guess it depends what you're thinking about in public health. So I think there are lots of opportunities. When you're looking at population health, obviously, you could pull out some markers that might be useful. So I think wherever you are in the world, there's going to be, different medications that are kind of commonly used, but there are some key key similarities between those. So I think maybe we're thinking more about kind of diagnostic genetics that might be used at a population health level and public health level, but because pharmacogenetics is really about predicting response to medications that you're going to use. So there could be some application. I just am not really sure how to further answer that question.
Yeah, I think that's a tricky one to foresee too, because I think that public health, the way that data is used in public health in the future will probably be very different than it is now, but it's hard to foresee exactly what will happen. I think one thing that's interesting and relevant right now is a lot of government organizations are trying to really change how they think about using data in public health as a response to the COVID-19, and trying to figure out ways to analyze patient information and see issues, detect these kind of population-wide issues earlier. So who knows? There could be a lot of innovation in the future in that area too. I think one of the big challenges is also around data sharing and how do you protect patients' privacy while using that information at a population level. That's a major challenge that's pretty hard to address.
Yes.
And if you're in the US, there's been recently some big changes in that in the legislation around how data is shared between organizations. But that's a topic for another day. So anyway.
Okay. So we've got a few more minutes left in the session here. We have a few questions as it relates to some more sales type things. So I'm gonna move past those ones. For those of you that have asked sales specific questions or questions about the product expansion, those sorts of things, the email that you'll be receiving after this session will include access to contact GenXys or DrugBank sales teams. So feel free to ask those questions in that way. We're going to move on now to a question from Sharon. What is the turnaround time for patients?
Yeah, so in terms of pharmacogenetic testing, I'm assuming. So I believe, so the testing right now is through Life Labs. So I believe it's 10 to 14 days. Although don't quote me on that, I'm not 100% sure, but it's 2 weeks basically, would be kind of the ballpark on that. So it's pretty quick.
And once you've done that test, you'll be able to keep using that for quite a while till you've updated the panel, right?
Yeah, exactly. So we keep our software very up to date. So if there's any changes in how you interpret the pharmacogenetic data, that's reflected in our software. So once you have the test done, everything's piped through automatically to the software. So there's no need to enter or keep track of any of that data, although you can have a paper report if people really want. And then, yes, we do keep them up to date. So recently there was a change in CYP2D6 phenotype definitions, and there was an international consensus on that. And so we are working on those updates. So we do try and keep everything as up to date as possible in the software. And the software is what enables us to do that. If you just had a paper report, you could send people multiple reports, but it would start to get pretty confusing.
Okay.
Great.
So we've got another one here from Thapello. As students studying pharmacogenomics, how do we figure out which drugs require pharmacogenomics testing and which do not?
Yeah, so in terms of requirements, so requirements are a bit different than recommended. So there are some medications for sure that have required tests. So for example, in the US, abacavir is required to have an HLA test before starting, and that's laid out in the product monograph and in some guidelines. There are some international guidelines that recommend pharmacogenetic testing. So for example, the American College of Rheumatology gout treatment guidelines recommend another HLA, which helps predict severe hypersensitivity reaction to allopurinol for certain populations of patients before starting that medication. In general, I mean, so PharmGKB is a great repository of pharmacogenetic information. So you can search on there by gene or drug, and that's a really easy way to find information about a drug if you're looking to see if there's any relevant pharmacogenetic information. And then also there are several guideline groups like CPIC that create guidelines for what you should test for that's relevant to certain medications, and then how to clinically use that information. And I think that's the important step is once you have that information, how do you use it? So we try not to test for anything that we don't really know what to do with. Or if there's not great evidence for it, we also generally might not include it because, if there's one study that comes out that says, oh, people with this variant need, lower doses of the medication. If we're then telling everyone to lower their dose of that medication based on that one study, and then another one comes out and refutes it, then you start to get into that, are you giving people the wrong information? So we really try and make sure that there's multiple studies supporting an action based on a pharmacogenetic variant before implementing it. But yeah, I think as a student, there are lots of sources. I would say probably the PharmGKB database would be a good place to go to look for information on genes that are related to a medication.
Great.
Yeah.
So we are at 45 minutes. We have quite a few questions left. Are you both okay to answer a few more?
Yeah, sure.
Okay, awesome. All right, so from Nuvik Lee, do you get feedback from patients about the results of the software's prediction?
So we do. So some, as I said, we've had some positive feedback about, medication options that were presented to them that they then, their clinician might have decided with them that, yeah, that was a good idea, and they tried it and they did have some success with that. And then, so that's one thing. We don't necessarily get feedback from everyone. We are looking at, as I said, using AI and using patient information that they can feed back in. So we're developing software that will allow patients to track, their outcomes when they take a medication, so that we can get more of that data back and really use that and kind of see what people kind of are having as a result of the medication that they take.
Okay, so we've got another one here from Adib. Can pharmacogenomics cause drug-to-drug interaction?
I think I kind of understand what you're asking there in terms of, so pharmacogenetic variation will affect how a drug is metabolized. So it actually potentially could affect another drug-drug interaction. If you have, say, drug A that's affecting the metabolism of drug B, and then you have a pharmacogenetic variant that affects drug A, you could have this kind of cascading effect where, more of drug A causes more of an effect on drug B, and that's a result of your pharmacogenetic variant. So there are lots of different ways that genetics can play into that. But I think what's often interesting is that drug-drug interactions are often mediated through those CYP pathways, and pharmacogenetic variations that affect drug response are often in those CYP pathways as well. So you can have a lot of interplay of drug-drug and gene interactions altogether.
Awesome. All right, the next question comes from Jeffrey. Similar to drug-drug interactions, are there ever any recommendations regarding dietary compound drug interactions like grapefruit juice and CYP3A4 drugs?
Yeah, that's a great question. So yes, grapefruit juice, one of those things we learn about in pharmacy school and everyone counsels on. So yes, I think generally we are looking at things like supplements as well when we're considering interactions. Right now we don't actually have food interactions in our software. I think that is generally more of a monitoring or counseling point once you decide to prescribe a medication and a patient fills it, in terms of spacing certain supplements away, or in the case of something like grapefruit juice where you are affecting a CYP enzyme. Perhaps if someone is unwilling to give up grapefruit juice, you might have to choose another medication and not prescribe something like simvastatin. So it is something we're considering for sure, and the DrugBank database also helps us pull the information about some of those supplements so that we can store them in our database if people are taking them. And then those drug-food interactions are definitely something we're thinking about in the future, but right now not a big part of the software.
Awesome. So we've got a question here from Rashmin. Hello, everyone. Can pharmacogenomics overcome the problem of idiosyncrasy?
Sorry, what was the question again?
Can pharmacogenomics overcome the problem of idiosyncrasy?
Oh, I see. Yeah. Well, that's the hope. So I think there's so much about the human genome that we don't fully understand. So right now we're picking out, these variants that seem to have a pretty big impact on drug response. But obviously there's, genes that affect those genes in terms of how they're expressed. So there's so much complex interplay between the genes in our body as well as the environment that we're in, or other disease states. So yeah, the hope is someday we would be able to look at someone and say, you are going to respond this way to this medication, or this is the best medication for you. So that's the dream. I don't know if we'll ever get, all the way there, just because of how complex the human genome and the human body is. But we're hoping to get close.
Awesome.
Nice.
Very exciting stuff. All right, David asks, how do you, in a sense, weigh the literature when attempting to prescribe treatments using pharmacogenetics? To expand a little bit, I am wondering how you would consider all the data that would come from preclinical studies maybe versus a phase 1 clinical study, or observations that are found in clinical studies, but not the major findings?
Yeah, that's a great question. So I think, so we do a couple different things when we're looking at literature. So first of all, we're not narrowing down on a single medication. Usually for patients, we're presenting kind of the best options from a safety and efficacy point of view, as well as kind of comparing the costs between them. So generally we're not looking at brand new medications that haven't been proven to be efficacious in the treatment of a condition. Usually we're basing kind of the backbone of our treatment algorithms on medications that have been established as, a standard of care, or, effective in treating the condition without kind of those major drawbacks. Generally, we're not looking at a lot of those early clinical studies yet, because for most of the conditions that we're working on right now, there's lots of really well-established medication treatment options. I think where the evidence gets tricky often is for the pharmacogenetic association with drug response, because sometimes you don't really have the clinical studies looking at, sometimes you don't have those clinical studies looking at for this variation. This is the response you get. Often there might be serum levels that you're looking at, and those are harder to predict. Does a 50% increase in serum level really have an effect on the drug down the road? And that's a challenge we have with looking at, kidney function, because those kidney function, testing and then, adapting medication doses, those are often done on serum testing as well. So I think that's probably the most challenging, rather than looking at those early clinical studies for drug approval. But yeah, generally we're looking kind of from the big picture down to the small picture with the pharmacogenetics in terms of how that affects medication response. But everything we use, we're evaluating for, through critical appraisal to make sure that the evidence we're incorporating is of the highest quality.
All right, so we've got two questions. One's from Anne. Is this applicable in the oncology field? And then kind of combining with RAMI, could it be used to predict drug-to-drug interactions and side effects in case diseases such as hepatitis or hepatic cancer?
Yeah, so definitely in oncology, it is used a lot, I think, already. Like here in BC, the BC Cancer Association does use pharmacogenetic testing. I think there's kind of two sides of that. So there are some drugs that you can test for your pharmacogenetic variants in yourself, and then predict some response to medications. But they also, in oncology, they also use a lot of tumor genotyping. So they're looking at the specific cells in the tumor to see what medications the tumor might respond to, or the cancer cells might respond to. So they use it in a bit of a different way. In terms of predicting drug-drug interactions, or in liver impairment, or liver diseases. I think that's really interesting because a lot of those drug metabolizing enzymes are in the liver. So there can be lots of effects. Usually what we're looking at is, if there is, severe liver impairment, that will often affect many different drugs. And if you have to adapt a drug dose based on liver impairment and you have a pharmacogenetic variant that also requires adapting the dose, generally we'd say you should probably stay away from that medication because there's likely to be safer alternatives for that patient.
Great. Awesome. So we have quite a few questions left. So I'm just going to pick a couple more to get us to that 10 o'clock mark here. I guess Alberta time, 10 o'clock. From Nambuso, does GenXys involve computational chemistry in terms of gathering data for your software?
So I don't think we have had anyone through computational chemistry work with us, no. So we involve a lot of clinicians right now. So in my team, we have several pharmacists and physicians involved. And then we also have consultants that we use for that information. On our lab team, we have lots of geneticists as well. So we've got kind of a broad spectrum of people, but yeah, no computational chemists yet.
And the next one is from Sharon. So I think this was a follow-up to a discussion that you were having earlier. But Sharon asks, because there are some FDA changes in the US regarding PGX, are the GenXys reports different than a Canadian report?
Yeah, so that's an interesting question. So there are definitely, some drugs that are available in the US that are not available in Canada and vice versa. But we do, so if we're reporting on medications that have recommendations that are just from the FDA, we do make that clear on the report. But a lot of the information that we're including on the report in terms of translation and clinical use of those pharmacogenetics are generally through international guideline groups or research. So they're less specific to a certain region. But we do make it clear if it's an FDA recommendation versus a Health Canada recommendation, especially in our software.
Perfect. So we've got one more question here from Roland. How often do you update information on kidney and liver function of patients, given that these can change at any time?
Yeah, that's a great question. So it's generally up to the users of the software to keep that updated. But in terms of EMR integration, which we're moving towards, that will allow that information to stay more up to date. So if we're able to link into an EMR and pull that information as it's available, that obviously is ideal. But that information can be updated anytime by the users of the software. So if patients are able to access their lab values, they can go on and update that. Or clinicians can also do that if they're using the software. So the answer would be we update it as often as they have information available.
Great. So I think that's all the time we have. So I want to say thanks so much for your time joining us today, Andrea. It was a lot of fun to learn about what you're doing at GenXys. And thanks to everybody who joined and all your questions. Yeah, it was our first Fireside Chat. And I think it went really well, and I certainly had a lot of fun. So thanks to everybody.
Yeah, me too. Thanks for having me.
Yeah. All righty. Have a good one.
Okay. Bye.
Bye.